Morning, everybody. Dr. Gillard here. It is week 9. It's Monday. You guys are on vacation today. I think it's Labor Day today. But I still have material that you are responsible for. And so here we go. This one will not be on the virtual classroom. This is only able to be seen here on my Professor Doug's lecture page. Or I may put it up on chirogeek.com. I'll send you a link to it, though, obviously, because that's probably how you found this. All right, so we're going to finish off talking about formation of the important neural tube and neural crest cells today. Here we go. Remember, last time we started talking about formation of the neural tube, and before it's a tube, it's a flat plate running right down the midline on the dorsal surface of the trilaminar disc, the brand new created trilaminar disc. And we said that certain biomolecules are released from the nodal cord, as well as the primitive node, the AVE cells. We'll see a slide on that. But these little biochemicals soak up into the regular ectoderm. Remember, I didn't partition this off, but this is all epithelium-like tissue, and it's called ectoderm. Well, the ectoderm in the middle gets these biomolecules soak into it and convert it. Specifically, turns off BMP, which we'll look at in a second. But they morph into a neural-type tissue. And so they're kind of a cross between ectoderm and neural tissue, so they're called neuroectoderm cells. Collectively, they make up the flat plate called the neural plate. All right, that's kind of where we left off last time. If you look at an overhead view, if we look through the amniotic fluid and we cut that away, sorry, I got a little frog in my throat today. Yeah, there it is. Over the top, we see the primitive node here, so it forms between the oropharyngeal membrane and the primitive node. Remember, the primitive node is slowly regressing, going backwards, but it's elongating the nodal cord as it goes backwards. And so our biomolecules are still being secreted and going to elongate this neural plate as well. And that's kind of where we left off on. So how do you actually make neuroectoderm cells? Well, as I mentioned, the trick is you have to turn off BMP4. Right? If cells secrete BMP, they become ectoderm cells. But if you can turn off the secretion, turn off the genes in the nuclei of these ectoderm cells, they morph into their default tissue state. And the default tissue state of these ectoderm cells is a neural tissue, and they call that a neuroectoderm. So that leads to the question, who turns off BMP4? Well, there's actually 3 inhibitors of that. Now, this is an advanced question. When you're studying for boards, this is probably one of the last type of questions, unless you're trying to absolutely crush boards and want to be in that top 5% of students. But the primitive node secretes 2 of the inhibitors, and these inhibitors are called noggin and cordon. And that's all we'll say. We won't say anything more about the mechanism, but they soak into the ectoderm cells. They get into the nucleus and turn off the BMP4 gene. Nodal cord, AVE cells, the anterior visceral endoderm, and even the precordial plate secrete Cerebus 1, which is another BMP inhibitor. So between those 3, the nuclei, the genes in the chromosomes get turned off. Now, we have another problem. So those ectoderm cells are locked together with E-cadherins, and so those need to be changed as well into one that is more amenable to become neural tissue. And remember, cadherents are what locked. Remember, specifically, cadherents make up maculoadherens and zonula adherens. Strong AKA for macular adherence is desmosomes. So those are no good for neural tissue. So remember, Dr. Doe, I'm sure, taught you that NK-adherents are what are used for cells in the brain. And so those need to be converted to NK-adherents. All right, we'll leave it at that. So what does this neural plate do? Well, it forms the neural tube. So what does the neural tube do? Everything. It's super important. It gives rise to our entire brain, not the entire brain, but most of the brain. Well, yeah, it does give rise to the entire. I mean, stuff can migrate into it. Neural crest cells migrate into it, but it gives rise to the bulk of the brain, as well as the spinal cord. And of course, there's the noodle, as I call it, up there, without the bones protecting it. Spinal cord is that white thing going right through the vertebral canal, which we've talked so much about in spinal anatomy. Right, so super important. There's always also some miscellaneous things, like important things, like the retina of the eye is super important. It gives rise to that as well. All right, so far we have this little plate here. But now the job, as the primitive streak regresses and the nodal cord elongates, we're going to make a key-like structure. So this neural plate is going to turn into kind of a key-like structure. All right. And so it's going to be wide. The original nodal cord, or the original neural plate, was wide up in the cranial end here. This is the head end, or the cranial end up here. Caudal end would be down here. And so it's gives rise to this key shape, and that's what we're making here. This is important. This is not happening. This is happening underneath the ectoderm layer. So ectoderm is removed here because we're looking superior to inferior. So that's important. That, of course, none of the board books tell you. Always confusing for anybody trying to study this stuff. So there's a process for this elongation, and it's called convergent extension. Convergent extension. And that's the elongation process. And that means new tissue is forming from the parent tissue. The parent tissue is that round plate up toward the cranial end, just behind the oropharyngeal membrane. Right? Why? How come it's key-shaped? And we won't get crazy into this, but the new cells that are forming as the nodal cord or as the primitive streak regresses and the nodal cord elongates, the new cells are intercalated. There are desmosomes, and the cadherens are really, really tight, and they pull the cells much tighter together than the original cells. And then they're converted, of course, into NK-adherens. But they're intercalated is the bottom line there. There's another word that we have to understand called the planar cell polarity pathway. This is actually what causes the elongation or the convergent extension of the neural plate. And so the planar cell polarity pathway uses certain receptors and biomolecules to allow the convergent extension process to happen. Who is used? It's a super complex pathway, but all I'll say is Wnt is used, and some receptors have to be made as well. This frizzled, Seltzer, Vangel. Those are the three receptors that are involved in the planar cellular polarity pathway. It's super complex. I tried to get into it before. It just got too deep into the weeds for, I think, these lectures are geared mainly for chiropractic students. So there's so much I really need an embryology part 2 to do this right. But we have only 1 hour a week, which is not enough time. So out it goes. All right, so now here's what we have. So far we have our original plate, and then we have this narrow extension. Notice how the primitive streak has regressed. and the nodal cord has elongated. Remember, the nodal cord is forming underneath this, and this plate. You know what? Did I say the ectoderm is? This is happening on the ectoderm here. Did I say that? I think I messed that up. This is happening on the ectoderm layer, right? So we haven't stripped away. This is not inside the mesoderm. So I think I messed that up somewhere. Where did that go? Yeah, this is all. This is the ectoderm is still in place. All right, sorry about that. Very early here in the morning. Brain is not full power yet. So this original plate region is going to form the future brain, and this narrower portion is going to morph into the future spinal cord. All right, so now we have a plate, a key-shaped plate made. How's that going to become the brain and especially the spinal cord, which is round? Well, we're going to have to fold it. We're going to actually create a tube out of that structure, and that's what happens next. So the very first thing to making a tube is we need to fold it. We need to start folding this thing. So about day 18. And before I start this is Carlson, which is my favorite embryology book by far. It's not a Board of Chiropractic Examiners book yet. I hope they adopt it. But he says that we don't really know 100% sure which one of these mechanisms work. I'm going to explain it as a single mechanism because it's easier, and that's what our chiropractic board books use. But there's probably other things going on at the same time, so it's not as simple as this mechanism I'm going to describe. There's numerous, as I said, mechanisms, intrinsic and extrinsic. But nevertheless, here we go. So the first thing that has to develop is a point where this thing bends. So we're going to turn our flat plate into a V structure. Flat plate, we're going to turn it into a V-like structure. That's the first thing that's going to happen. And in order to do this, we need to take these cells right in the middle of the plate, and instead of rectangular in shape, we need to make them triangular in shape. If we make these cells triangular, this thing has to bend into this formation. That's exactly what happens. Right? So the triangular shape, some of the ectoderms in the very midline start to become triangular in shape, and that causes us to get a triangle like going here. That's a cool triangle there, but it's a little bit, that would be upside down though, really. I would have to turn that upside down, but I couldn't do it because that's such a magnificent historical monument, I guess you could call it. It's the pyramid and the Sphinx, of course. I guess I should really turn it upside down, but oh no, I guess that works. Yeah, that works. So anyway, brain's still not woken up yet. So let's take a look at how this works. So if I blow up that flat plate, and kind of like that silly picture I drew, you can see that the apices of these neuroectoderm cells have scrunched together. And the base here, down by the mesoderm, hasn't done anything. So if you scrunch these down into a triangular shape, it has to bend that flat plate down. That's exactly what happens. So I know you're all wondering, well, what biomolecule is responsible for this triangular shapedness? And it's an easy one to remember. It's shroom. And again, it's a little more complicated than this, but the key biomolecule is shroom. And shroom is released from nodal cord and from the primitive node, and it soaks into these cells, and it causes the apices of these cells to scrunch together. Specifically, remember, cells have like a cytoskeleton, right? And so the microfilaments of the cytoskeleton pull in and contract, and that's what causes that triangular shape. All right, so now we have a V-like structure, and the point in the midline at which these folds occur, that's got a name where the first bending occurs, and that's called the median hinge point. Median hinge point. I like that word. There should be more stars on this slide. The newly created walls now, because instead of a flat plate, now we have a right and a left wall. Those are called the neural folds. Neural folds. And the neural folds create a valley, and that midline groove or valley is called the neural groove. Neural groove. So if this picture shows up on the test, you would know these parts. So the median hinge point would be not medial, median, meaning in the midline. That would be the median hinge point. This would be a lateral fold. This would be a lateral fold. And the valley created here, that would be the neural groove. Got it? These are special little cells here. If you're wondering what these cells are, they're still neuroectoderm, but they're going to become one of the most crazy, mystifying, I don't know if it's mystifying, but they're just a miracle, these cells. These are going to become the neural crest cells, which we're going to talk about after we get through this. Now, the other thing is the neuroectoderm is kind of helping this folding. The neuroectoderm senses, this is regular ectoderm. The ectoderm is going to help this folding process. They sense that they've lost some of their brethren. And so, according to them, there's a big hole here, and that's got to be filled. So these guys split via mitosis, and split again, and split again. And so we have cells growing, ectoderm cells, run-of-the-mill ectoderm cells growing in this direction. And you can see what that's going to do. It's going to, I don't have a three-dimensional model, but if we are increasing the size and growing these cells this way, this thing has to bend downward. And so this inward growth of ectoderm cells is assisting the bending and the formation of the neural tube here. All right, that's everything I said. And there's just an example of that. Eventually, this is going to completely fill in this region, and the ectoderm is going to be whole again. And that's when the neural tube is going to actually dive down below the ectoderm. And that's where I got messed up, because the neural tube is below the ectoderm, but the neural plate forms as part of the midline ectoderm. Okay, everybody good with that? Lateral hinge points. So soon, this doesn't look much like a tube, right? That's a V structure. How are we going to make that a tube? Well, what if we make some more of those triangular, I'll exaggerate these, but what if we make some triangular cells here again? What if we call out shroom again? And that's exactly what happens. We get some lateral hinge points as more shroom hits the midpoint of the lateral walls. And it's the same story. The cytoskeleton shrinks at the apices of these cells, makes them triangular in shape, and now we got a lateral hinge point here. And pretty soon, all of these cells are going to become triangular in shape, which is going to form a nice tube here. All right, and now you can see that also the ectoderm cells have elongated. So this space is getting closer. These cells are still growing, trying to close the gap here. And these weird cells right at the very edge, I guess you would call it the dorsal surface now. Here they would be at the lateral surface of the neural plate. Now they're at the dorsal surface of this neural plate, which is folded. These are magical. They're getting closer and closer to each other. So about the same time, cells at the junction of the ectoderm and neural plate start morphing. And then now we're talking about these neural crest cells. This is really, really important. These neuroectoderm cells, I've said several times, many authors call this the fourth germ layer because it creates so much tissue and so many neural structures. So I completely agree with that. We'll come back to those neural crest cells. You're not going to like those neural crest cells because this is a first quarter class, and they create many second quarter structures that you'll learn in gross anatomy too. Make sure you put, when you're doing your student assessments, if you haven't done those yet, it's really good to put that this course really should be in second quarter after we've had gross anatomy 2, which it really should be. Why they put it in first quarter, but I'll tell you, because I did medical school, I did a few quarters of that, or semesters, and sure enough, embryology was quarter 1, 2. I think they just want to, I don't know about physical therapy school, but I would bet it's probably in that as well, first quarter. But nevertheless, let's create this neural tube. Let's keep the story going. So about day 21, right? So that's what? That's the end of week 3, the starting of week 4. All the triangular shaped cells and the hinge points and the growing ectoderm finally connects the once flat plate into a tube-like structure. And they fuse together, and now we have a hollow neural tube. We should also, we'll talk about next week, the formation of the somites, which are super important. They're mesoderm clumps that are so important for so many things. This somite story is starting about now as well. So we got a couple stories going on at the same time. Right? So here's an example we've said before. The cells are a lot of them are triangular shaped, and we have a pretty round looking neurotube here. But notice one really important thing. Our little dark green or green color cells here right at the very, well, now it's the dorsal surface of the old neural plate. They are going to morph into a new structure called the neural crest cells, and they're actually going to have the ability to migrate. And it's a complicated story. I won't get into it, but they can move, and they do move through a complicated system. It's more than just pseudopodia, too. But they morph into tissue, and they get the heck out of there. They don't want to be trapped in this neurotube. And right after the neurotube, or right after the neural plate finally closes to form a tube, they hang out right here, which is very short-lived. They immediately split in half and migrate right to this region here where they stay. And man, they migrate everywhere in the body. We'll look at some of the places they go, but that's where they're going to go shortly. Notice how the ectoderm is now like nothing ever happened. It's reformed. And yeah. All right, neural crest cells, again, the dorsal region, or that end part of the plate, in order to leave, they have to become mesenchymal tissue. And so here's the next tissue. We've talked about epithelial to mesenchymal transformation, right? When ectoderms, or when epiblast cells move down through the primitive streak, they have to become mesenchymal tissue. Right now, in fact, we have mesenchymal tissue still. The mesoderm is mesenchymal tissue. It never turned back into epithelial tissue. And we said the hypoderm, or the hypoblast, was converted into a epiderm or a hypoderm. And those cells turned back into epithelial cells. Remember the cells, well, I won't go through that again. I'm going too far in the weeds. We've already covered that stuff. But the point is, we've seen epidermal to mesenchymal transformation again, and that's what has to happen to those pre-neural crest cells. And once they do that, they're called neural crest cells, And that, again, they migrate out of the closing neural tube. So this has a name, of course, this getting out of Dodge, so to speak, if you're a cowboy movie fan. Get out of Dodge. This escape from the folding neural plate is called delamination. Delamination. And as we said, they typically hang out right above the newly formed tube, and then they migrate down to those dorsolateral corners, as I already drew. And so we've done this a couple times. But this process of them escaping again is called delamination. Are we good? All right, so jumping ahead in time a little bit, when is the process of neurulation over? So this closing process, the whole tube doesn't close at once. It actually starts more in the middle region. And interestingly, the middle region is going to become the cervical region because we have the head region, which is more cranial. But nevertheless, it doesn't close at once. It closes like a zipper, cranially and caudally. And so that open part, some of the tube is a closed tube, but some of it is still kind of a V-like structure. And so those open ends have a name, and those are called the neural pores. Specifically, we have a really large cranial neural pore. Why is it larger than the caudal neural pore? Well, because we have a plate up in the cranial region, a much wider neural plate, and then we have a skinnier neural plate. Remember the key shape. So it makes sense that's much larger. Some authors, Carlson calls them the anterior neural pore and the posterior neural pore. So medical students who use that book, that's an AKA for that. We've explained why it's cranial and anterior are AKAs because of the folding process. Cranial region will remain the cranial region, but it folds to the anterior. That's why they use anterior. That's always confusing. So here now, this picture is where because remember the ectoderm is back. So this is going on underneath the ectoderm. So the ectoderm has been removed to see this picture. But here we have a big open region right here as this folding is still occurring. And so that's called the cranial neural pore. And you can see day 21 doesn't look like much, but by the end of the 4th month, it looks like this. It's almost completely closed, and the caudal is almost completely closed. Here's these somites that we started talking about. So these are already, that story's already going on, and we'll get that next week. Neural pore fun facts. So the cranial neural pore closes first, about day 25. Yeah, you can also say the 18 to 20 somite stage is day 25. Caudal neural pore is the very last to close, about day 28, or the 25 somite stage. Once that closes up, the process of neurulation is officially complete. And now your central nervous system is a tube. Some wicked birth defects happen if it fails to close. And I took a bunch of slides out because, again, we're getting too deep into the weeds. But this is a really important slide. Gross picture warning. Sorry about that. I can't see my slides coming. Here is a human fetus that didn't make it. And it shows us what happens if the neural tube or the neural plate or that V structure fails to close. In fact, this one never closed. Up in the middle back and up in the skull, never closed at all. And so when the key portion of the neural plate fails to close, that's called a rachis. Rachis. Super high board yield for medical students and chiropractic students. Even more so for spine. Anything with a spine, brain, chiropractic boards love that kind of stuff. I have a feeling. And if the skull fails to close, it's called a cranioscus. Cranioscus. It's almost like saying just kiss, to ray kiss, and cranioscus, or kisses. It's really more like kiss to say it right. But anyway, there it is. And this is rare. You can see it like this. But usually you see just the rachis. Maybe it's just this much has occurred. Because you can be born, and this, of course, is a spina bifida, which they will talk about in neurology and neurophysiology more. But got it? Rachis is when the spine is left open. Cranioscus is when part of the skull doesn't form and you can see the brain. Both of these are no good, right? All right, now let's get into these neural crest cells. Again, called the fourth germ layer. They're so important. They arise from neuroectoderm cells that were kind of modified at the last second. What induces these neural crest cells to become neural crest cells? Well, it's bone morphogenic protein gene they have to be exposed to just the right amount of BMP. Remember, the BMP is turned off. We had to turn off BMP4, right, to make those genes. We had to turn off those genes. And it really is not exposure. It's really the genes. Just a little bit of BMP4 needs to be expressed in these cells to make it work. And so let's talk about that. So ectoderm cells in the midline were exposed to BMP inhibitors, right? Remember that, chorda, noggin, cerebrus 1. So we had to turn that off. If BMP4 is turned off, it defaults into a neuroectoderm structure, right? But remember, the ectoderm over in this region, right? So this thickened up to form the neural plate. This is a kind of cross-sectional view here. We turned off BMP4 here. BMP4 is cranking. It's still being produced by genes. These genes are still turned on, so that keeps it ectoderm state. But this region right here is interesting. You have to have just some of the BMP4 genes. And it's more complicated than this. I'm watering this down a little bit. But the gist of it is the BMP4 genes have to be expressed, but just a little bit, like a medium exposure or medium secretion of BMP4 is going on here. And that makes them special. And that's not an easy thing to do. So what induces neuroectoderm cells to become neural crest cells? They have to be expressing medium levels of BMP. All right? That will make them morph into these neural crest cells. Now the question is, well, how do you do that? How do you make BMP? I could be putting BMP4. Probably not for our tests. I think BMP is probably good enough. But you should know BMP4 too. I guess I should have been consistent about that. So there's some transcription factors that need to be turned on in order to make this work. So we have MSX, DLX5, and PAX are the transcription factors that you need to have. These genes also need to be turned on in these cells. And these transcription factors turn on other genes like FOXD3, SOX10, EST1. Drives you crazy, doesn't it? That's the nerd alert. And with a combination of the receptors and these other gene products turned on, you get these mesenchymal cells are created, which are called neural crest cells, special mesenchymal cells. Note in here for chiropractors, the Moores, which is one of the chiropractic board books, says that Wnt and beta-carotene signaling is also involved in this process of creating the neural crest cells. And specifically, these activate a gene called the GBX2 homobox gene. is turned on, and that's also an essential gene. They know because mutations in any one of these results in a non-viable fetus. And again, don't freak out about this. That wouldn't even, if you're just trying to do well on the boards, and you can still do really good on the boards without memorizing all this crazy stuff. This is probably low yield, but for those of you, especially medical students, who need to go a little deeper into this, there you go. How do neural crest cells break loose in order to migrate? Well, all the cadherins have to turn off, right? Because remember, we had N-cadherins and maybe a couple E-cadherins in these outer cells, these cells that are going to become neuroblasts. They might have some N-cadherins and E-cadherins. So we won't go make this crazy either, but there are some more gene products that they get exposed to, call them the Cam-Turner offers. That's snail 1 and 2, a new one that we haven't talked about, twist and FOXD3. So those are you want to create a mesenchymal cell in this region. Those are what have to be turned on. All right, so now they're mesenchymal. They can swim. They can move. So what's next? They need to migrate, and they're mesenchymal cells. And mesenchymal cells can migrate. So they've already went through this is kind of out of order. They've already went through epithelial to mesenchymal transformation. That slide can be worked on a little bit, but now they can migrate. All right. They're going to migrate great distances, as we're going to see. And they need a gene product that protects them specifically from, if you're going to be if you're going up to the brain, or you're going up to the face region to create the dermis of the face, you don't want to stop in the neck region, or you don't want to stop in the chest region. So premature stoppage is a big problem, and there's all sorts of birth defects that occur when these don't migrate to their needed location. And we have a protector that helps them, and we won't get into how it helps them, but the SOC10 gene. The SOC10 gene is what has to be turned on in these mesenchymal cells in their nucleus, and that SOC10 product is the protector of them. So it'll make sure that they make it to their predestined location. Mutations in SOC10, all kinds of fetuses, all kinds of defects in the fetus usually won't make it. Let's see, and there's even deeper we can go. So before they start moving, there's some other guidance, ligand receptor pairs that have to be turned on that help guide them to where they're supposed to go. We have a protector, but how do they even know where to go? So the robo slit, and the ligand would be robo, the receptor would be slit, but the receptors are also made by genes. It's just a protein, and the ligand is also made by genes. So these genes have to be turned on. Robo slit, neurophilin, and semaphorin, and ephrin, and EPH all have to be turned on. Moore's, the new 2020, which I just got at the start of this quarter. I've been looking at that. Looks pretty much similar to the old Moore's, but there are some changes. So Moore's also added the FOXD3, snail2, SOX10, SOX9 in there as well. Pretty low yield stuff, I think, for at least chiropractic boards. But nevertheless, for those of you who have a photographic memory and can memorize stuff like that, there you go. You can see it's an incredibly complex process, and we still don't even understand the whole thing. So where do they migrate? Right now we have a trilaminar disc with with a notochord. Primitive streak is almost gone by this point. We have an ectoderm, a mesoderm, endoderm. What are the pathways that they migrate? Like pioneers in covered wagons heading from the East Coast, exploring the West. That's what these things are. They're migrating. Where do they go? So they tend to migrate along the surface ectoderm, specifically surface ectoderm has now formed a connective tissue layer underneath it, a basal lamina. So they like to migrate through that basal lamina. And the neurotube also, those cells have created a basal lamina. So you could say they like to migrate along the basal lamina of the surface ectoderm and neurotube. The board question could say that where do they like to migrate? Maybe they'll just say through the surface ectoderm. They migrate along the neurotube or along the surface ectoderm. Maybe they'll leave basal lamina out. They have to avoid some biomolecules like chondroitin sulfin containing proteoglycans. They have to watch out for that type of tissue. That'll freeze them, so they have to steer clear of that. That's where our SOC comes in. Integrin proteins are activated. We'll just leave it at that, which also help with their journey. And we'll just leave it at that. They're more of a motility type thing. Now, this is, out of all the stuff, I mean, everything is important, but of all the stuff I've talked about, this is going to be really important stuff. All medical boards, to my understanding, and chiropractic boards, they all like to, and I've said this before, they all like to ask you, what was the embryological start of this tissue? Where did your skin, the epidermis that you've talked about in dermatology, where did that come from? Well, that comes from the ectoderm. Right? What about the next layer down, the dermis? Where does that come from? Most of it comes from various parts. We'll talk about how mesoderm breaks down into 3 components. But it comes mainly from mesoderm, but not all of it. The skin on your face and neck actually comes from neural crest cells. That's the kind of stuff that you need to know. So let's go through what these neural crest cells become. When I think neural crest cell, I think nervous structure. And it's not complete, but that's what the first thing that pops into my brain. So they basically form, with some exceptions, they form most of the peripheral nervous system, all the nerves, sciatic nerve, all the peripheral nerves. They form many of the sympathetic nerves and parasympathetic nerves. The ganglia of nerves, of axons, they form most of those as well. The sensory ganglia of peripheral nerves, which is most like the very important dorsal root ganglia we've talked about. Disc herniation touches that thing. The patient usually has horrible pain, a very sensitive structure. And those discectomies don't work out as well because the surgeon has to monkey around with that. And it's a sensitive, that's where the cell bodies are located. The ganglia of cranial nerves, like the Gasserian ganglia, aka semilunar ganglia, aka trigeminal ganglia, pops into my mind. Those are all formed from neural crest cells. Sympathetic ganglia. Watch out, I left this one in here because this is an instructive little section. But the older board books, and they've changed this now, but they used to say Merkel cells. Like in 2010, Merkel cells were derived, like Langman said, Merkel cells were derived from neural crest cells, and that's no longer true. But I left it in here anyway because we need to talk about this. Is always confusing. Now they're derivatives of primitive epidermal cells make them. But then Merkel cells are part of something called the Merkel disc, right? AKA Merkel cell neurite complex, Merkel disc. And if you haven't studied this yet, it's an important structure. So let's take a look at it. So here's a Merkel disc. Let's get the big picture first. So here's a cross-section of your skin. So the skin has 4 main layers. If it's globurous skin, like on your palms or on the soles of your feet, you have 5 layers. The rest of your hirsute skin, your hairy skin, has 4 layers. The main layers are the stratum corneum, or the horny layer, the stratum granulosum, stratum spinosum, And then down here, we have a single layer of cells called the stratum basale, made of basal cells, spiny cells, granular cells, and horny cells. Or corneocytes is another AK for that. If you're in the palms, like this diagram was in the palms, you got this extra layer called the stratum lucidium. So here is our Merkel disc, and it's really important for light touch, for picking up a pencil, knowing how much pressure, for identifying. If you're blind, it's incredibly important. But it's discriminatory or light touch. If you close your eyes, how do you tell the difference between a pen, like I'm just holding a pen in my hand, and your iPhone? So that's what it's important for. And it's made of two parts. There's the Merkel cell, and then there's a nerve that connects to it. That's called the alpha-beta afferent neuron. And those two components join right in the stratum basale. So they're super. These Merkel discs are really, really high-yield board stuff to know, I think. All right, so let's go back to what do our neural crest cells morph into. Well, peripheral, we said kind of, did we said? Yeah, we said penis nerves already, but even some components of the peripheral nerves. So Schwann cells for the large, fast bundles of axon called nerves. Because remember, nerves, when I say nerves, I'm really talking about bundles of axons that have action potentials zipping in both directions. But Schwann cells, they produce a myelin, a kind of a coating. It's like the insulation of a wire in a way, but it allows for the signal to travel not in a continuous flow, but in a jumping manner. And it makes the signal, the electrical signal, go very, very fast. Schwann cells are a class of cells called glial cells. I don't know if we'll get to that this quarter in spinal anatomy, but I do have a video on that. You'll get that, though, elsewhere. The glial cells are another super high yield, all the different glial cells. The neural lemma, which is the coating around, it's like the cell membrane or the plasma membrane around a Schwann cell, is also derived from neural crest cells. Here's another. I like to ask questions from this. We got to do a little neurology to get you ready for the real neurology classes. Here's a nerve, run-of-the-mill peripheral nerve. So you have to be careful when you say the peripheral nerve is derived from neural crest cells, because not all of it is. The axons and things like that, and the myelin is definitely derived. The Schwann cells, here's one cell, there's its nucleus that wraps itself around in these little sections, and that is a Schwann cell. There's gaps called node of Ranvier or node of Ranvier, whichever tomatoes. But this is covered by a plasma membrane, so the Schwann cell and its plasma membrane are both made by these neural crest cells. But interestingly enough, here's a run-of-the-mill nerve. It has the classic three coverings. It has the epiderm or the epineurium. That's the thick covering around the so-called nerve. Inside the nerve, you have bundles that are bundled together by another tissue. That's called the perineurium. And these are called fascicles, perineurium bundles, fascicles. Fascicles are what made up of axons. These are the wires where the signal actually travels down. If you pull one axon out and take a look at it, it has a covering called the endoneurium. So all of these tissues were derived from mesoderm, not neural crest cells. So the only thing that's derived from neural crest cells, you have to unravel. You have to take the skin off the actual axon and look at the skin. Yeah, it's okay to call this whole thing an axon, I think. The axon is actually the shaft in the middle. These are the coverings of the axon. But if you take the outer covering off, the endoneurium, sometimes called the sheath of Henle, now we see the coverings of the actual axon itself, and they're Schwann cells. And that is what's derived. Who makes a Schwann cell? Neuroectoderm cells make a Schwann cell. It also makes the neurolemma around it. Okay, are we good with that? The autonomic system, so we're back to what do neural crest cells morph into, the ANS. I think we said that already, but let's look at it a little more deeply. Sympathetic chain ganglia. Okay, we already said ganglia, but celiac ganglia is also derived from neural crest cells. The parasympathetic, not the pre, but the post-ganglionic neurons are derived from neural crest cells. Parasympathetic ganglia, you'll learn these next quarter. The submandibular ganglia, we talk about that in fifth quarter because that can be confused sometimes with a submandibular lymph node that's swollen up. Sphenopalatine, the ethmoid, and the ciliary ganglia all were made from neural crest cells. How about the skin? I teach dermatology, so I do like the skin, and I've been using examples to get this in your brain. So the skin is interesting. It's the largest organ of the body, and it has a double embryological organ. The skin is technically two layers. It's the epidermis and the dermis. That's the skin. The hypodermis, which has all sorts of AKAs, subcutis, subcutaneous, superficial fascia, those are all AKAs for that third layer of tissue down. That is not considered the skin, according to the chiropractic board books anyway, so we don't include that in the skin. But ectoderm is easy. The skin that you feel everywhere on your body right now, your face, your legs, your arms, your hands, that was derived from good old-fashioned ectoderm. Simple as that. Nothing more to say about that. The next layer of the skin, the dermis, is a little more complicated, as we'll... Let's see, did I talk about... I have a slide, so I won't spoil that surprise. Oh, I guess I spoiled it already. The dermis, everywhere in your body, the dermis is created by mesoderm, either intermediate mesoderm or lateral plate mesoderm. And I don't think the boards are going to get that deep, but maybe they will. But we'll talk about lateral plate mesoderm and intermediate mesoderm and paraxial mesoderm in the next lecture. But the trick, the fly in the ointment with regard to the dermis is that the skin of the face and the skin surrounding your neck is actually created, the dermis of the face and the neck is actually created by neural crest cells and not mesoderm. So that's a tricky little area. That would be a nice little board question. So make sure you understand that. It also creates a lot of times the board books or the embryology board books just call them pigmented cells, which is, you got to do better than that, I think. Pigmented cells, it's not even an official AKA. They kind of make that up. But we're talking about melanocytes. Really, melanoblasts give rise to melanocytes, but melanocytes are created by neural crest cells. Melanoblasts live in the stratum basale layer, and their job, they're like little, here's one here, it's like a little octopus. Its job is to inject all the neighboring keratinocytes that were formed with melanin. And melanin, depending on how many melanocytes there are and how active they are, that's going to cause your skin to be various shades of darkness. African Americans have a lot of melanocytes, and they're very active, and therefore their keratinocytes are heavily injected, and they're very dark because of that. And maybe people really white, like myself, I'm pretty Caucasian, pretty white. I don't have that many melanocytes. That's a good thing and a bad thing. That's why white people get cancer more than African Americans get cancer, because we have less melanin. Melanin tends to go over the nucleus here and protect the cell against UV damage. So the darker your skin, the more protected against cancer. I'm already starting you first quarter. I want you to look at this lesion. If you ever see a skin lesion like this is actually a dermoscopic view. But if you ever seen a lesion that's variegated, bigger than the tip of a pencil eraser, like a number 2 eraser. This is melanoma. This will kill you. This is malignant melanoma, which can metastasize very, very quickly. So that's a mutation of these melanocytes, and we'll talk about that more in 7th quarter. Some miscellaneous things. We're still talking about neural crest cells and what they can become. The word leptomeninges you should be familiar with. Hang on a second. Specifically, first of all, what are leptomeninges? That's pia mater and arachnoid mater. That's in the brain. It's located in the brain region. So the leptomeninges, pia and arachnoid of prosencephalon, which is the forebrain, and the mesencephalon, which is the midbrain, guess what? Derived from neural crest cells. The adrenal gland, specifically the adrenal medulla, is also derived from neural crest cells, as well as the most important cell of the adrenal medulla, which is the chromaffin cell. Chromaffin cells are super important because they secrete epinephrine, aka, what's the aka for epinephrine? Adrenaline. So that's really important. Carotid bodies, which you'll learn about, type 1 cells specifically, are also derived from neural crest cells. When we get up into the skull, now this is where we start to leave the kind of the neurology part. Neural crest cells are responsible for formation of bone, specifically the squamous portion of the parietal and the squamous portion of the frontal bone. Those are the main portions of the parietal and frontal bones. So part of the calvarium, or the brain case, is derived from neural crest cells. The maxilla and mandible, those are huge bones. Those are derived from neural crest cells. And bones of the facial skeleton, you could say in general, are derived from neural crest cells, like the ethmoid bone, the lacrimal bone, the little tiny 3 bones, the auditory ossicles, all derived from neural crest cells. And by the way, I think we do this in lab tomorrow, but you need to know, next quarter, you're going to get into every nook and cranny of the skull. Got some really popular skull videos where I will go way deep as you could possibly go into every nook and cranny of the skull. But this is going to be on my test. You need to at least learn the basics if you don't already. So we have a frontal bone here, parietal bone, derived from neural crest cells. These are the squamous portions. Sphenoid bone is right here. It's like a little creature. You can look at my videos, but that goes inside as well. Temporal bone, zygomatic bone, mandible, maxilla is all here. Nasal bones, ethmoidal bone, could have added that. And this drawing isn't the greatest. Or are they trying to, that's actually the ethmoid bone is here. This is the nasal, this is the lacrimal bone right here. It should be much skinnier than that. And that's the ethmoid bone. That should be longer than that. So this artist is off a little bit. But those are the main bones that I want you to know. There's the auditory ossicles. You probably know these already from undergraduate. But there's the hammer, or the malleus is here. There's the incus is here. So sound is coming that way. And then the stapes is right here. Okay. Derived from neuroectoderm. So that's enough of that. So how do they know what to become? How do neural crest cells migrate to the ethmoid region to become the ethmoid bone? How do they know that? And we're not 100% sure on that. But there's 2 kind of camps. One theory says that all neural crest cells, they have equal potential to become anything. And it's all what makes them become ethmoid bone, as opposed to dermis of the neck, is the region to which they migrate into. So it's the chemical soup of the area they migrate into, which tells them what to become. So their differentiation completely depends upon the biochemical environment into which they migrate. Another school of thought says that's not true. The neural crest cells are pre-programmed to swim up to the ethmoid region of the facial skeleton and become the ethmoid bone. They know that before they even embark on their covered wagon journey. And so most authors say it's probably a mix of these two theories, which I would agree with. We did this already, I think, but let's finish our neurotube story, because remember, the neurotube doesn't form over the sacrum and coccyx, right? So we have to actually go through a process called secondary neurulation to make that happen. So let's talk about that. Secondary neurulation, we did already, I think. So secondary neurulation occurs underneath the tail bud. We talked about the tail bud was how the primitive streak was degenerating, and that very last bit of the primitive streak down into the caudal region created a clump of cells called the tail bud. And that's where the story starts. So sacral and coccygeal neural tube begin with structures with tissue forming underneath the tail bud. And the first thing you get is something called a medullary cord, which becomes hollow. So that forms beneath the dorsal ectoderm of the tail bud. Forms when mesenchymal cells migrate out of the tail bud. Just like they were migrating out of the primitive streak, they migrate down through the tail bud region. Their genes are manipulated to become this medullary cord. So here's a picture. Here's dorsal, ventral, would be there. And there's the neurotube, but it stopped. There's the L5 region. So there's no neurotube in the sacrum and coccyx. So the tail bud kind of showed up in this region right here. And cells from the ectoderm or epiblast, depending on what stage they're in, migrate through here, and they gather here to form a tissue. And that tissue is going to become the medullary cord. And guess what? We're going to join that to here. But that's second. This is the process. Primary neurulation stops right here. But this continues. This is secondary neurulation. Okay, after the medullary cord is formed, it hollows out into a cavity. And now we have a central canal running through. We have a hollow tube, just like the primary neurotube is. And now we just have to connect them together. And that's exactly what happens. We won't talk about the chemical soup, the genes that are turned on to make that happen. We'll just say they connect together, and now we have a complete neurotube. Secondary neurulation is complete. After secondary neurulation is complete, the neural pore is closed down there. Remember, that was day, 28? So, yeah, neurulation is complete. And, yeah, so now we have a tube. So what? Well, the next thing, what happens next? Well, we need to start forming that brain, and I'm not going to go crazy. I don't have time. Well, hopefully the neural anatomy and neurophysiology teachers will get into this, a little of this. But I'll start you off. We are going to form the primitive brain. And the primitive brain is just made of neurotube. Remember, it's wider up in the top because the neural plate was wider. Those are going to morph into 3 sections. And that's the primitive brain. So here's our neurotube. You could call it the spinal cord if you want. It's still the neurotube. And here's the much larger kind of cranial portion of the neurotube, and it's morphed into 3 sections. So this is the primitive forebrain, a.k.a. prosencephalon. You need to know these like the back of your hand until you get out of school. Then you probably will never use them again. But as you go through neurology, and that's always the hardest part, most challenging part of the boards, is the neurophysiology, neural anatomy. So yeah, that's the prosencephalon. Mesencephalon will be the midbrain, and then the hindbrain is called the rhombencephalon. So know these three. Guarantee you there's a perfect question for me to ask. Right? Board style questions have 4 answers, choices. So that's a perfect one for me to make some weird word up and see if you can pick those out. Further subdivisions. So as time goes on, we can subdivide 2 of the sections. So the prosencephalon will divide into this green and blue region. So we have the diencephalon here in green, and then we have the telencephalon or the telencephalon, tomatoes, up here. The mesencephalon doesn't change, but the rhombencephalon morphs into 2 parts. A metencephalon, not to be confused with the mesencephalon, and the other portion of the rhombencephalon that manifests is the myelencephalon, which will, of course, become the brainstem. But I won't go any further than that. Diencephalon is important to me, though, because I teach endocrinology, the more advanced endocrinology, and the hypothalamus is in this region, and hypothalamus is very important for ADH and CRH and lots of good stuff. Did you... I hope I was able to mute that cough out. I like this Yeti mic. I can mute things really quick. Got a much better sound, too. Before we dig any deeper, there's more to the story. How does folding occur? We're going to talk about folding in the next lecture. Actually, yeah, that's the next lecture. So we'll talk about somatogenesis, the forming of the somatomeres and somites. Super important lecture. The formation of mesoderm into paraxial mesoderm, intermediate mesoderm, lateral plate mesoderm. And then we'll talk about folding. We got a busy lecture next time. It's our last class. All right. Hope you enjoyed it. See y'all later.